Peter's gone through what heritability is, how to calculate it through solar, and how to set up solar. My talk's more of an application that we've done through the Enigma Consortium, using solar both for variance components models like heritability and also going into specific SNPs, looking at single SNP fixed effects, but not just in one site, doing a sort of multi-site joint analysis, looking and comparing both meta-analysis and mega-analysis, which Peter explained a bit. So how did we use solar eclipse? Again, we're doing a joint analysis of imaging studies specifically to improve the power for discovery. You might have seen Derek Hybar's talk yesterday, but the goal of Enigma is to kind of pull together all these different data sets to improve the power of genetic discoveries and finding variants that actually affect brain structure. And Peter and I both lead the Enigma DTI working group, so we're focused on looking at white matter changes and the genetics of white matter. So we'll go into a little bit about how to create a pedigree matrix for mega-analysis, specifically for imaging, and then compare the mega and meta-analytical approaches for using solar. So for those of you who are not familiar with meta-analysis, basically the definition of meta-analysis is a quantitative statistical analysis for several separate but similar experiments or studies in order to test the pooled data for statistical significance. And this is actually a little bit old, but just a couple days ago I checked Google Scholar to see how many articles in 2015 already, in the past couple weeks, have been published on meta-analysis for human genetics, and it's over 1,300. And at this very conference last year, at this time, it was closer to 1,200. So the more and more meta-analytical studies are being done. And so far, in addition to looking at brain structure, it's been very important for discoveries in all sorts of diseases, cardiovascular diseases, cancer, Alzheimer's, schizophrenia. But our focus here, as I mentioned, is the meta-analysis of imaging genetic studies. And I won't really go through much of this again, but just to kind of summarize Derek's talk yesterday, we've pulled together data from, I think he said over 40 data sets to look at the genetic effects of brain structure, especially subcortical volumes. But what about the brain's white matter microstructure? So these are the myelinated axons that connect the gray matter regions and connect the subcortical structures to the cortex. And they've been important for neurodegeneration and psychiatric disorders. And they depend on many complex processes and are highly genetically influenced, and we'll get a little bit more into that. So what Peter and I have been focusing on is using DTI images. So diffusion-weighted imaging is MRI-based, as I'm sure you are all very familiar with it. But the simplest model that you can get from the diffusion-weighted images is really this diffusion tensor images, because you don't need a lot of directions, and very many clinical studies are also able to image patients in a short amount of time with a few directions, and you can reconstruct a tensor at every voxel. And from this tensor, you can calculate the FA. And that's basically is an estimate of to which the water diffusion is directionally constrained. So you know whether it's anisotropic or it's isotropic in a particular voxel. And the higher the FA, the more anisotropic, and the closer it is to 0, the more isotropic it is. So at every voxel, we have a measure of FA. And through Enigma DTI, we've been able to pool all this data together. Although there have been several papers published on white matter heritability and disease risks and how they associate to FA measures calculated through DTI, there haven't really been, these are all in single sites. So a lot of these genetic variants that have been maybe found in one particular data sets to affect white matter structure haven't really been replicated at least not very often. Usually these are single-site studies. And again, we want to, the white matter structure brings in a sort of different element than the subcortical structure. So there's a different genetic component variance that than what's already been done. So here I'm showing the genetic correlation between various DTI regions and the subcortical volumes. And you see the shared genetic effect really only goes up as high as 40%. So other than that, the brain structure of the white matter and the subcortical volumes are pretty genetically unique, so it's really important to study both. There is also genetic overlap between white matter and psychiatric disorders, both bipolar, major depressive disorder, and also schizophrenia. And we can, so this kind of motivates us to look into it further. There have been a couple full GWAS studies of DTI. However, none of them have really come up with anything genome-wide significant. And that's the 5 times 10 to the -8 threshold, which we use for the entire human genome for essentially testing for 1 million SNPs for the for any correction. So how do we do a multi-site DTI GWAS? Well, we want to do it, again, because I mentioned, to increase the number of subjects. But would a track from one study necessarily be the same as a track from another study? And would the tracks actually be reliable the same amount across the different studies? There's so many different added complexities of DTI that, so like the different voxel sizes can greatly affect the FA that you get, or the number of directions where the image, how the image was acquired can also affect the FA measure you get. So how can you pool all this data that was collected in all sorts of different ways together to see if something is heritable, and to find such small genetic effects? As we found out, the effect size of a particular SNP on human brain structure is about like 0.5 to 1%. So how are we going to find such small effects with such diverse data? So first we want to know, is it heritable? And this is where solar comes in. How can we find out regions of the brain that are uniquely heritable, or I guess not uniquely, but like together they're heritable across all the different data sets, no matter how the image was acquired? So what we're doing here is both a meta-analysis and a mega-analysis, and we've combined many different family studies. So large pedigrees, as Peter first talked about, and also a couple twin studies, so you only have monozygotic twins and dizygotic twins. And here you're just looking at the difference, looking at the correlation difference between monozygotic twins and dizygotic twins. And if the monozygotic twins would share 100% of their genome, or are significantly more correlated than the dizygotic twins, then you can estimate that the trait is heritable. So we looked at 5 different cohorts here, all in with several hundred subjects, all different ranges. GOBS was the large pedigree study, so we had subjects from age 19 to 85. QTIM was a very narrow-aged twin family studies. They were all in their 20s. And a couple of adolescent cohorts, and NTR was also a larger study of different age twins, so they were about 18 to 45. All these were done with different scanners. The field strengths varied differently. We had 4T data, 1.5T data, 3T data. Voxel sizes, again, completely different. Basically 2 millimeter cube to 1.7 and 3 millimeter thickness. And again, the directionalities are completely varied, from 55 directions, 94 directions, to a more standard 32. So how can we combine all this and actually run out of solar? So the first initial steps we developed in Enigma DTI is to use some very basic image processing methods. We created a template from various sources of data, from a lot of high-resolution data scans, to map everyone into the same space. We used a lot of the tools developed at FSL using TVSS particularly to skeletonize our template and make sure that all cohorts were able to register their subjects to this template and project their FA values onto that same skeleton. And we partitioned the skeleton into different regions of interest from previously defined ALICE. So for creating the pedigree files, Peter did go over this a little bit, so I'll just briefly run through it. For the pedigree, we have the IDs, the father's, the mother ID, all separate, and also the genotype ID. I guess this is the I forgot what this one was, but the pedigree number, because we had multiple families in the same pedigree file. And then the twins, again, we had to again do the IDs for both the subjects for which we had the scan, their mother and father, for so the solar would know to make sure that the families were correct. And then again, all the standard information. We also had an added column for monozygotic twins to make sure that solar does read these as those that share a kinship 100%. So for the mega pedigree file, we can essentially take those two files and merge them together. Now the only added column we'll need is class. So if we had 5 data sets, we'll have these numbered from 0 to 4, and each being a different data set completely. And we wanted to not only do this in regions of interest, which are very similar to things that Peter has already described, but also wanted to do this voxelize. And we made use of the loaning pipeline to split the images and kind of create individual solar files. And this was done before the nifty reader that Peter just talked about. So what we developed here is this thing that kind of looks like a squash spider or something, but there's essentially three modules here that do 3 separate tasks. And I'll talk about this a little bit. So the first one, once you have it set up, it's fairly straightforward, and then you'll know exactly what to change. At first it looks a little scary though. So basically you keep all the subject's information in an Excel file that you can save as a .text. And then you call what you are interested in by the columns, just like in the pedigree files. And then you can tell the pipeline kind of what you talked about in the question and answer session. You can tell the pipeline how many nodes you want to use up, and that it knows how many pieces to split the image up into. So, so here's kind of a basic example. This is your spreadsheet that you feed in to the information file. It looks into the spreadsheet, which has your family ID, your subject ID, the zygosity, and the path to your images that you're interested in, and all sorts of variables that you may or may not be interested in. You can have as many as you'd like, and you just kind of tell this pipeline which ones to call. So you give it a massive file so it knows which voxels to look at in these images, and how many covariates you're interested in, age and sex. You can also do, we're not showing it here, but you can also do combinations of the different variables. So if I was interested in the interaction between age and sex, I could model that as well, without having to add in an extra column. For example, if you've gone through and looked at your images and noticed that a couple of them are bad and you don't want to include them, but maybe you want to keep them in the spreadsheet for other reasons, you can kind of filter out the ones that you've QC'd and said, okay, this one's not good, so you can add in a filter. And here you're saying that you have 100 nodes, so it's going to split up all your images into 100 pieces and run heritability on that. So here you essentially put in the covariates in how you want to write and how you want it to run. So I'm interested in doing just a linear effective age, but also age squared, and all that interacted with the effective sex. So I'm looking at age, age squared, age by sex, age squared by sex, and also sex. And then this is what splits it up into the 100 pieces. And then you can tell Solar which command you want it to use. So if you want to just run the univariate heritability model or for each individual site, or if you want to run the mega analytic file, or run all the sites together. And that there are other options as well that we can add. And these were all the output files of the model. So it looks at every voxel individually, as Peter mentioned, and it saves these into a temporary file, which then which each print the heritability, the standard error, the significance, and any other variable that you want Solar to output. And you can take these, and in the last node, it combines them automatically, the temporary files, and gets rid of them, and then you have your nifty image. And this is a voxelized skeleton FA heritability map. So that's kind of what it looks like. And here we've shown how we did run heritability on each of the different cohorts at each of the various ROIs. You can see a lot of them vary significantly. So for example, the cerebrospinal tract, some of them have a fairly high heritability of, it looks like 0.6, while others, the standard error actually included 0, so it wasn't heritable at all. However, other regions, like the body of the corpus callosum, was extremely heritable in all five sites. And this is, when we did it voxelized, it's just showing 2 of the sites. This was the twin study, and here you have the GOB study, the large pedigree study. So we also talked about mega-analysis. So if you want to analyze everything together, you can tell Solar that you want to do the mega-analysis, and what it'll first do is take the FA distributions of each site, regress out the covariates at that site specifically, and do the inverse normalization for each site again separately, and join that into a distribution for everyone, and do the heritability analysis as a group entirely. So for the meta-analysis, we haven't really covered it that much yet, so I'll talk a little bit about how we ended up doing that. We took the individual outputs for each of the cohorts that we ran the heritability analysis on, and there's no standard way of doing a meta-analysis on variance components-based factors, which heritability is, but we looked into 2 different methods for weighting the heritability at each site before we pool them together. One is weighting each site's effect by the number of subjects that were included in that site, and that's the kind of the n-weighted heritability estimate. And the second estimate is a standard error weighted estimate, or really looking at weighting by the inverse variance. So as we saw, solar does output the standard error of heritability as well as the heritability estimate itself and the significance value. So we can use this to weight each site's heritability before we pull it all together. And then you can get an overall variance estimate of the heritability across all the groups too, so you'll know whether this is actually significant across everyone or not. Because you could get a high heritability value, but if the variance is too large, of course, it'll include 0 in the confidence interval, and it won't be heritable. And we can use those same estimates to calculate a significance value based on the standard error and heritability. As I mentioned, there's really no standard for doing this, so we did explore both methods. We essentially convert the heritability to be a beta value for the effect size, and use standard methods for both the n-weighted analysis and the inverse variance weighted meta-analysis to get the z-score and convert the z-score to a p-value. So here are some voxelized results. So we have a meta-analysis weighted by the number of subjects at each site. You see that's slightly lower than the meta-analysis weighted by the standard error at each site. And then the mega-analysis is a kind of a more smoother estimate. You can see the voxels kind of blend in nicely together with all 5 groups together. And these are 3D versions of the corpus callosum. So again, comparing the voxel-wise results, we had in the meta-analysis of, I know you can't really see, but this is the meta-analysis of standard error on the y-axis, and then the meta-analysis with the n-weighted on the x-axis. And we have some values where the standard error weighted version was estimated a lot higher than the meta-analysis, but otherwise they were very collinear, and I think the correlation coefficient was 0.87. And same thing with the mega-analysis, the standard error weighted one. There's a few regions that were really noisy, and the standard error weighted version gave it a really high estimate when neither the n-weighted or the mega did. But we're looking into why that's the case, and I think we figured out why, and we're fixing that. And the meta-analysis with the n-weight and the mega-analysis was also highly correlated. This is if you're just looking into all the ROIs, it might be a little bit easier to see. The n-weighted was always a lot lower, and the standard error and mega-analysis were very similar across the board. All of them showed really low heritability estimates for the cerebrospinal tract and the fornix. So those regions, we decided that the heritability estimate is kind of low or inconsistent across the different sites, so we don't think those are good for pursuing a genome-wide genetic association. And here is a forest plot of the 5 different sites and their heritability estimates for the average FA across the entire skeleton. And here we're showing the different pooling effects for the n-weighted, the standard error weighted, and the mega. So now that we've done that, we want to know how robust these estimates actually are. What if we removed one site from each analysis? How does that compare to the full thing? And then how do the different meta and mega analyses compare? So each one of these columns of 3, I guess, is removing one site. You can see that for the most part, we kind of kept the same pattern throughout. So So these are all fairly robust estimates. I mean, so mega does look really good, but how practical is it, especially for genome-wide estimates? Not everyone can share the data. There's obviously a lot of IRB issues. There's a lot of people just don't want to share the genetic information, especially because it is sensitive. You don't want to know who's been in your study. You also don't want to share the data. So it might not be as practical. What if we could do a partial mega-analysis and a meta-analysis? So here we're showing different combinations of using the data and how it compares to having the entire data set and looking at the mega-analysis and different meta-analyses, like mega-analyzing 3 and meta-analyzing 2 into that estimate, or groups of 4 and 1. Anyway, we tried all sorts of various combinations. With every combination, you can see that the cerebrospinal tract had a very low estimate. The superior longitudinal fasciculus, that's the SLF right here, is pretty much high, very high, and in almost every permutation of the data. But otherwise, across the board, the correlations between the meta and the mega analyses were really high, actually, between 0.8 and 0.81 and I think 0.97 or 0.98. And overall, the meta-analysis, so with all 5 of them meta-analyzed together, was correlated at 0.81 with the mega-analysis. And in terms of their significance for heritability, they all were really significant for the most part. So what we can take back from that is that all results using the mega-analysis and the meta-analysis are fairly stable, and you can combine data sets that you have all the information for with data sets that you might not have the information for and still get really reliable estimates. And there might be benefits to that using a partial mega over doing an entire meta-analysis. But there are questions that remain in terms of the genetic interpretation of this. So what does it actually mean to combine heritability results from different populations if heritability is supposed to be a population-specific measure? And therefore, is mega-analysis even appropriate for this? But for our case, because we want to use this as kind of the basis and the foundation for why we want to do a genome-wide analysis, we do find this is actually very meaningful, and we want to look into the regions that are heritable across the board and are influenced by genes. The same across various populations, various data sets, and various groups of people around the world. So now that we looked at heritability, what about fixed effects? As we talked about briefly before, there have been many SNPs that have been associated with FA and DTI measures. But again, it's always been like a single site study, and there haven't been many replications. And we want to look into the meta-analysis of this and see, are we missing anything if we do meta-analysis versus mega-analysis? And is the mega-analysis always more powerful, especially with fixed effects, which is what our overall goal is. So we're looking into this a little further now. So in the last week, this has actually increased quite a bit, so these are very preliminary results. We now have, I think, 12 or 13 sites with over 6,000 scans, and we looked into a few candidate SNPs that have been previously published and to see whether these replicate across the different sites and whether pooling the data and pooling the information actually improves power or not. So the largest single ethnicity group we had was made up of over 3,000 CEU subjects. Some of the groups we talked about were from our previous study with heritability, and they were family-based cohorts, which you might not usually see with SNP studies. But a lot of others were also unrelated samples. So we actually took family-based studies, twin studies, and unrelated samples in solar, and were able to model this. This is just kind of a breakdown of the different groups, where they all came from, several European cohorts, a couple Australian cohorts, and several here in the United States. So the SNPs we looked at specifically, a couple were from the genome-wide searches that were done on FA measures. There were 3 Alzheimer's disease SNPs. Again, these had all been published before to have been associated with DTI measures. We initially had 11 SNPs that had been previously associated with psychiatry, but then one of them ended up having a really low minor allele frequency in most of the groups, so we had to take it out. And here we go, this one had a very low minor allele frequency, so several groups didn't even maybe had 1 or 0 people with both minor alleles, so this was not practical. Thank you. So this is the results comparing meta-analysis in blue and mega-analysis in red of, the only one I'm showing here right now is the average FA, so FA across the entire white matter skeleton. Here are, this lower bar graph is the p-values, but that's not really what I want to focus on. Essentially, some of them were below the 0.05 or uncorrected threshold, but if you're going to correct for genome-wide significance in this sample, they haven't reached genome-wide significance yet. But what I really want to focus on right now, which I think is important to note, is the differences in the error bars of the meta-analysis and the mega-analysis. For several of them, you'll see that maybe the mega-analysis had a higher beta value, so this is the effect size. Either direction, this is positive effect, this is negative effect with respect to the minor allele. But you'll also notice sometimes that the mega-analysis standard error is actually quite high and includes, oftentimes includes the 0, where the meta-analysis might not have. But so like right here, the effect is slightly larger with the mega-analysis, but so is the standard error. And when we looked into this, particularly with the whole average FA, the mega-analysis standard error was always significantly higher than the meta-analysis standard error. So this kind of reminds you to take caution when doing mega-analysis and make sure to look into the confidence intervals and not just kind of look at the effect and say it's bigger. So to break it down by the different sites, the top candidate SNP was the BDNF SNP, and we note that I think this is kind of interesting. These 2 groups, Imagen and NeuroImage, were below, that the p-value was closer to significance. These were both the adolescent samples, so I think between 12 and 14 for both of them, while the rest are mainly adults. So this was in the CEU population only, and this is kind of everyone together. Both of them, the p-value was not terribly different. So now we know a little bit more about the kind of compounds of the meta-analysis and the mega-analysis, and different things we kind of need to be aware of. But we do need more groups to join for the large genome-wide meta-analysis, and whether we can do a meta-analysis and a mega-analysis together. We can definitely do that if people want to share the data. We will do the mega-analysis in part, and obviously anyone who is not able to share the data, we currently our setup is the meta-analysis. So we can find important, hopefully discover more genetic variants that affect the brain and see if these ones that we have been studying are kind of global and across the entire, are able to be found regardless of the population that's being studied, and also how the images are acquired. I think that makes a big difference. So this kind of all falls into the Enigma DTI paradigm. We have a set of pre-processing suggestions. I mean, it is very data dependent, but we are providing several quality control measures, how to kind of create your FA maps, to look at them, make sure it looks reasonable, do the registration to the template and the skeletonization, do several layers of quality control. Extract the average FA within the regions of interest, and kind of make sure your data is normal, and do the genome-wide association. So a lot of times you can pick up outliers. We have noticed you don't need too much of the inverse normalization with these FA measures, although a lot of times it is useful if you're looking at the FA across the population of your cohort. You'll notice that for the most part, different regions of interest are almost follow the normal distribution, and if they're not, you'll be able to pick out some outliers in your data and see if anything went wrong in the processing. So that's basically the summary of what we're doing so far in Enigma DTI and in joint collaboration with Solar, and to kind of look into these joint analyses and how to use it on a voxelized level on different regions of interest, and how we can make best use of the power that we're going to have in this study. Another application of Solar that I kind of wanted to point out is something we just did with Peter a couple months ago, and this is a vertex-wise analysis of shape. I know there's a brief talk about shape analysis yesterday, so I thought it would be appropriate to bring it up. But basically what we've been doing in a lot of these subcortical regions is to take the extracted volume and kind of map it to a sphere that we have a template for, and then we can map the template back to the shape. And here you can get an estimate of how thick each vertex is compared to like a medial curve. And here we ran this shape analysis on all the subcortical structures in both HCP and RQTIM data. So we had over 1,400 subjects in this heritability analysis using twin data. It was all twin data, and we ran it through the Solar Eclipse pipeline that was modified to take in these vertex files, and we found really high heritability in almost all the shape regions. I think there's a little bit of the amygdala that was not significantly heritable, and this color scheme is showing the lower bound of heritability. So if you take the h² value and subtract out and look at the lower confidence interval bound, that's what you're seeing here. So just to thank everyone here, Peter, who's been leading the Solar effort and promoting all these different ideas to test in Enigma DTI, and also Tom, John, Bennett, Paul, and David, who's been leading the Enigma DTI group. Thanks.
Generate heterogeneity statistics for across sites when you combine I², Q².
So we can look into like we haven't really looked into that heterogeneity with the meta-analysis, but when comparing the fixed effects model with the mega-analysis, when we see that the standard error is actually a lot higher with the mega, we know that there is quite a bit of heterogeneity within the DTI especially. I think the image acquisition parameters make it just inherently more heterogeneous than volume would be. Yeah, so that's why we kind of looked into this to see are we doing the right analysis or not, and we do think the meta-analysis is certainly a safe gold standard to use.
Yeah, I think the main problem there, I mean, there was a great hope to take the GWAS significant SNPs and to demonstrate how great they work for the megagenetic analysis, and what we ended up facing with.
We have a lot of things we need to clean up.
No, I think what we ended up facing is that it's difficult to replicate GWAS significant hits because, you know, the effect size is so modest, and it appears that the effect direction is variable from cohort to cohort.
Right. So we're hoping that increasing the sample size a bit will help determine one way or the other whether these could be really pulled together or not.
Yeah, but I think the real lesson here is that GWAS results, even significant GWAS results presented in a single cohort, may or may not be all that meaningful when applied to different cohorts, because had they been meaningful, or had they been, really, significant, like this heritability, we have very nice, we have no issues with heterogeneity across cohorts, with the same heritability of the same thread. Now, when it comes to, significant hits, we actually observe that it's not at all.
Yeah, it's very dependent on the cohort.
It's very much cohort dependent, and the alarming part is actually seeing the trends, at least